pleasure to have this opportunity to share this topic with you today. My topic is a blueprint for high-throughput screening using organoids. I'm Chen from ALIT Biotech company. And today I'll share the four main parts to you. Firstly, I'll have a brief introduction about organoid background, and then I'll have for the high-throughput screening, I'll share our ALIT total solution, and then I will focus on the following two parts: HIVITA for organoid perfusion modeling and chip development, and CASTR S2 for high-throughput imaging and analysis. Market survey show that the drug-related applications accounts for the largest proportion, 37%, and all of these show that the booming global market for organoid research, especially in drug screening and testing. Organoid, especially patient-derived organoid, reflects the characteristics of primary tumor and can accurately predict patient response to drugs. Now we have several models for drug screening compared with 2D cell line and animal model. Organoid, organs on a chip, and also organoids on a chip can have many advantages over them and show a great potential in drug discovery, screening, and precision medicine. This is a very common process for high-throughput drug screening using organoids, which can be divided into three parts: simple preparation, organoid modeling and development, high-throughput screening. Common well plates are usually used for organoid culture, high-throughput drug screening. In addition, more researchers using organ-on-chip for in vitro model construction and drug screening, but the basic process is simplified. However, there are many challenges. The use of traditional well plates results in lower cell viability, low outputs, and due to poor nutrient exchange within the droplets and poor consistency of organoid culture, a large number of manual fluid exchange operations and culture steps are difficult to standardize. When using in vitro chips for organoid modeling, biophysical environments is the most major challenge, which involves a large number of manual operations and personalized process within low outputs. In high-throughput screening assays, imaging analysis is one of the key methods. Microscopy methods with fixed focal length imaging can lead to unclear autofocus image, which can affect the analysis results. In addition, there is a lack of high throughput and accurate image analysis tools and methods, as most of the current image analysis tools are developed based on 2D cell line, which are not well suited to organoid analysis needs. Our ALIT Biotech company is founded in 2005 and had its headquarters and R&D center in Shanghai, China. We develop and manufacture innovative CASTR brand cell analyzers, including our regional viral series products and CASTR, and also have the HIVITA brand for organoid culture and system and chips to meet the rapid growing demand of cell-based research and therapeutic development. We also started a new company, ALIT Biotech, in the US this year, and this is the contact information for our US director, Dr. Yi Zhang. To address the current challenges, our ALIT company can provide a solution for high-throughput organoid modeling and drug screening. For the traditional 96 well plate for drug screening, cells first seeded directly into our organoid factory chip, and then put into the HIVITA system for automatic perfusion culture and rapid expansion. Compared with the line perfusion method in traditional well plate, it can save much time, great increase in cell vitality, up to 24 samples in parallel for high throughput expansion and biobanking. For organ-on-chip method, HIVITA as an open biomimic platform and multi-chips compatible enables automatic and consistent perfusion modeling and drug dosing in high throughput manner. Combined with imaging system and AI image analysis algorithms, CASTR S2 is capable of detecting a single 96 well plate in 4 minutes, and light standard AI models are available to ensure more than 80% accuracy. Overall, we provide a simple, more efficient method for high-throughput modeling and screening using organoids. Firstly, I will introduce how HIVITA system can be used for rapid organoid expansion and organ-on-a-chip development. The HIVITA system consists of two parts, the controller and our culture chamber. The temperature and gas control module make the system environment very stable. The fluid control module can adjust the pumping speed within the range of 1 microliter to 7 millimeters per minute. There are 6 pumping units in each chamber, so you can try 6 different perfusion parameters at the same time. Our software system meets the requirements of compliance management. All experimental information will be recorded. HIVITA system is essentially open platform, not only for our designed organoid factory chip for better organoid perfusion culture, but also compatible with a variety of commercially available microfluidics chips and organon chips, allowing users to do their own research and chip development. In HIVITA system, single chamber can achieve 6 organoid factory chips, up to 24 samples parallel testing, meeting the needs of high-throughput organoid culture expansion and biobanking. The compliant software enables fully automatic monitoring of multi-parallel testing and process development. Data shown that 15 microliter per minute is the most suitable flow rate for intestinal organoid expansion, and the cell vitality is greatly higher than in well plates. And our designed organoid factory chip has 2 longitudinal semi-permeable membrane, realizing the dynamic renewal of culture fluid without damaging the fragile organoid samples. This membrane design also ensures organoids can be real-time imaged. At the same time, the fluid shear given by perfusion can more simulate the in vivo physiological environment. Combined with the HIVITA system, they can achieve long-time continuous automatic perfusion culture without manual fluid exchange, which saves more than 67% time compared to the traditional well plate culture, and greatly improves the cell vitality in 1.5 to 30-fold increase in cell vitality than those in plates. And several areas of organoid bimodal distribution in this show nuts was greatly improved by using the HIVITA system, indicating it can greatly improve the consistency of organoid size distribution and ensure the reproducibility of downstream experiments. And this is an example for one of our customers. Cells from primary tissue were seeded into our organoid factory chips and then placed into the HIVITA system for continuous 10 days of perfusion culture. And we can see that the organoid in HIVITA system were larger and in better condition compared to the traditional well plates, proving that organoids in HIVITA system show higher expansion and success rate. As an open platform for perfusion culture, commercial microfluidics chips can be connected to HIVITA system and reservoir through common connector. Up to 24 samples can be used for high throughput, which can greatly save the traditional steps of using syringe pump and manually operation, and makes reproducible high throughput chips development, testing, and drug screening possible. Here is also an example. Self-designed chips or commercial chips can be plugged into the HIVITA system, and then high throughput imaging analysis can be performed using CAST-S2 system. Entire perfusion control and drug dosing process in the microfluidics chips can be automatic, controlled, and recorded. Up to 24 chips in parallel for high throughput testing. Recording of parameters and controllability of conditions ensures reproducible modeling process and repeatable results. Here are also the results of fluorescence viability analysis using CAST-S2 imaging system after drug testing with commercial chips by our customer. It is possible now to get the high-definition fluorescence imaging of the entire chip chamber. And after this stack imaging, 2D projection and staging, we can clearly see the overall organoid status after drug treatment. So next, I will introduce how to use CAST-S2 for rapid high-throughput imaging and AI-based data analysis in detail in high-throughput drug screening using organoids. Again, there are also many problems with current conventional imaging and analyzing methods, including out-of-focus image, lack of high throughput, and inaccurate analytical tools and methodologies. So in response to these challenges, our ALIT company have developed a new system called CAST-S2, specifically for high-throughput imaging analysis of organoids, equipped with 2 objectives, 4 times and 10 times. A cold color camera has 3 fluorescence channels, blue, green, and red. And it can achieve a very fast and automatic Z-stack imaging, 2D projection, and staging using artificial intelligence image algorithms. CAST-S2 can provide a very fast and accurate image analysis for quantitative characterization of organoid morphology and phenotypes. Also, the interactive approach based on AI model training allows for customizing new assays to meet the challenges of high-throughput assays of complicated samples. So it's not a traditional image system anymore. With this advanced image system, it can achieve a very rapid Z-stack imaging and 2D projection. It only takes 4 minutes to finish the bright field imaging in 1960 wild plate. And it has 3 kind of fluorescence image channels, blue, green, and red. And also with the automatic workstation, it is ready for high throughput automation, compatible with various consumer modes, can be selectable freely in the software, including from 6 to 384 well plates. This is a basic process for the CAST-S2. Firstly, it performs a Z-stack imaging and 2D projection at each position, and then it will move to the next position to capture the Z-stack imaging, and the system then automatically performs image staging to obtain, for example, the entire mesogeal dome image, and uses AI algorithms to perform image analysis and finally outputs a quantitative visualized image for display. All of the above imaging and analysis process were performed automatically by selecting a standard assay. For both bright field and fluorescence images, our AI algorithms can enable to perform accurate analysis and provide multiple morphological quantitative data, including organoids number, viability, total area, average diameter, and relative fluorescence intensity, and so on. CAST-S2 has standard AI algorithm models built into the software, which is currently compatible with more than 9 common organoid types, including colorectal cancer organoids, hepatic and gastric cancer organoids. Recognition accuracy can reach more than 80%, and some of them can reach 95%. In addition, we can also develop the customized AI models for new organoid types or complex experimental requirements based on AI model annotation and training data, and outputs the new methods for diverse analysis, including bulk analysis, single organoid analysis, and time course analysis. And then finally to form a new standard assay for customer to use. For example, the single organoid growth curve, label-free organoid drug response test. Currently, we can also provide this customized analytical assays service for free. So to summarize the CAST-S2 features, compared to traditional methods, CAST-S2 is capable of imaging and analyzing a 96 well plate in 30 minutes with bright field imaging takes only 4 minutes. This is a time saving of over 80%. Our software currently provides standard AI algorithm models compatible with more than 9 types of organoids, high accuracy, and we are also continually upgrading our AI models and will make them free to our customer. We can also provide custom development services based on AI algorithms to meet our clients' complex samples or new requirements. Again, all free. So with this high speed, high throughput imaging, high level accuracy, identification, and customized analytical assays, Custom S2 makes high throughput screening using organoids more simple and more affordable, and can greatly accelerate frontier organoid research and the development of organoid industrialization. Here is an example. Hepatic organoids from the 96 well plate can be continually imaged and analyzed in the software, and the quantitative and analyzed data and growth curve of the hepatic organoid morphology are available. Bright field imaging and analysis of each 96 well plate can be completed in 30 minutes. So with an automatic orchestration, it can up to 48 plates per day can be finished. Here is another example of high throughput drug screening using Cast S2. After drug treatment, bright field and fluorescence images can be captured, and organoid viability result after staining of the fluorescence dye AOPI can be directly used for subsequent decision making. And currently, patient-derived organoids for in vitro killing experiments, which are widely used for tumor immunology study, functional evaluation of in vitro killing efficiency of target drugs, and cell therapy drugs, using Cast S2 to perform organoid killing experiments not only provides the images of morphological changes, but also accurately identifies and quantifies analytical data, including organoid viability, immune cell viability, and the relative fluorescence intensity of organoids, making complex analyses that are not possible with traditional methods. Now it is possible. So finally, here in summary, our ALIT company can provide streamlined solutions for high throughput organoid modeling and imaging analysis in drug screening by using our organoid factory in Habitat system for continuous perfusion culture, and for the final high throughput imaging and analysis in Cast S2 imaging system. That's all. Thank you for your time and listening. And please feel free to contact us, ask questions, or contact us for the Habitat and Cast S2 prototype trial. Thank you.